All right, so for today's deep dive, we're jumping into the pharmacokinetics of the mRNA vaccines, the BNT162b2 and mRNA 1273 shots.
Yep. And the general consensus, the one backed by all the regulatory data, is pretty clear.
It's that they deliver a temporary set of instructions, almost like a prodrug.
Exactly, a temporary software update for your cells, and then it's cleared out of your system, mostly by the liver and other enzymes, within, a few days to a few weeks.
That's the baseline. But then you have this one study that came out by Mordechai et al. That made a really extraordinary claim.
A huge outlier. They said they found vaccine mRNA in blood, in placental tissue, even in semen, for over 200 days.
200 days. I mean, if that's true, that changes everything we thought we knew about how these things work.
It would. So our mission here is to really pick that study apart. Is this real biology, or is it just an artifact of their methods?
Okay, so let's start right there. With the methods, what tools did they actually use to find this?
This is where it gets really interesting. They used something called nested polymerase chain reaction, or nested PCR, and they pushed it to its absolute limit.
How so?
They ran 50 total amplification cycles. 50. 20 in the first round, and then another 30 in the second.
Wow, and 50 cycles, that's the big red flag, isn't it? Because in a clinical setting for like a diagnostic test, anything over 35 or 40 is basically considered noise.
It's indeterminate.
Precisely.
When you crank the sensitivity that high, you become overwhelmingly susceptible to something we call carryover contamination.
So you could be picking up just dust.
Essentially, yeah. Aerosolized fragments of DNA from a previous experiment run in the same lab. At 50 cycles, you are almost guaranteed to amplify these tiny ghost traces. It looks like a positive result, but it might not reflect what's actually in the sample.
Okay, so the method is already on shaky ground. What about the journal it was published in? The venue?
Yeah, it's the other part of the puzzle. It was published in the Annals of Case Reports by Gavin Publishers.
Yeah.
And that's widely identified as a predatory publisher.
And what does that mean in practice?
It means the peer review process is questionable at best. And you can see it in the timeline. The paper was received on October 7th.
Okay.
And it was accepted on October 13th.
6 days. A 6-day turnaround for a study that claims to overturn the entire field of mRNA pharmacokinetics.
It just suggests it completely bypassed the rigorous technical validation you'd need for such a massive claim.
So how does this outlier stack up against all the other validated high-fidelity data out there?
Well, the contrast is stark. The quantitative studies show that the lipid nanoparticle, the little fat bubble carrying the mRNA...
The delivery vehicle.
Right. That's cleared very quickly, mostly by the liver and spleen. The mRNA itself has a functional half-life of just a few days. The absolute credible upper limit for finding tiny fragments in the blood is about 28 days.
But hold on, I have seen some high-quality studies that detect fragments for up to 60 days in certain places. What's that about?
But that's the key distinction. That 60-day detection is localized. It's found in the germinal centers of your lymph nodes. And that's actually a feature, not a bug.
Yeah.
That's where your body is building long-term immune memory. It's not systemic circulation like the outlier study claimed. It's functional persistence right where you want it.
I see. And what about those really sensitive claims they made about finding it in reproductive tissues, the placenta, semen?
Again, the robust quantitative studies show zero evidence of mRNA transferring into placental tissue or cord blood, and the blood test barrier seems to hold up just fine. Large studies have found no negative effects on male fertility parameters around 70 days after vaccination.
So when we put this all together, what's the final verdict for the listener?
At the end of the day, it seems overwhelmingly likely that the 200-day persistence claim is a technical artifact. It's a result of an overly sensitive qualitative method, that 50-cycle PCR, published in a journal without proper peer review.
So clinical practice and policy should stick with the validated data.
Exactly. The validated data confirms the transient nature of these vaccines. But this whole thing leaves you with a really important question to think about.
What's that?
When you come across information about a medical breakthrough or a scary new finding, how do you tell the difference between a sensational outlier and a genuine discovery? Is the best first filter to look at the rigor of the methods they used, or is it the reputation of the place where it was published?